It's almost time for GCSE Biology Paper 2. So how confident are you feeling about describing the sampling required practical? Hi, my name's Dr. De Bruin. I'm a GCSE and A-level science teacher, and I also make revision videos on YouTube. 15% of all the marks in this exam paper are going to be for required practicals. And the good news is, if you're taking combined science, there are only 2 of them. But the problem is, when you did this one, you were probably so busy having a nice time out on the field that you didn't really do it very well, and you don't really remember all the detail. So the point of this sampling required practical is to figure out how does a plant grow differently depending on an abiotic factor, like how much light there is or what is the pH of the soil. You're going to use a quadrat, which is a square frame where you can either count the number of plants inside, or if it's got intermediate divisions, you could even do percentage cover. And every time you put it down and count the plants inside, you're also going to measure whatever that abiotic factor is. So maybe you have a light meter for measuring light, or maybe you have a pH probe that you're putting into the soil. Now there are 2 options for how this can work. One is that you've got 2 different sample sites. Say Farmer Nige has got a really acidic farm and Farmer Al has got a really alkaline farm. In that case, you want to do lots of sampling at both sites, but not the entire farm. That would take you all year. So you divide up your sample space to make a kind of grid, and then you use coordinates to assign numbers. You open up Microsoft Excel, other spreadsheets are available, and use the random number generator to tell you which bits you're actually going to sample. So you maybe do 10 at Farmer Nige's site and 10 at Farmer Al's site. Calculate some means, jobs are good. The alternative, particularly if you're looking at the amount of water there is, or sometimes the amount of light, is that you want to do systematic sampling. This is where you take your tape measure, a really big tape measure, and you make a really long track called a transect. So maybe one end of your transect is right by a river where the soil is really wet, and then you're moving away from there and it's getting drier and drier. And you maybe say, I'm going to sample every 10 metres. So every 10 metres, you put down your quadrat, you sample the plants that are inside it, and you take a reading of how wet the soil is. So with this data, you're not going to take a mean, because you don't want to know what the average number of plants in your transect is. You want to know how is it different at the very wet end compared to the very dry end, or at the very dark end under a tree compared to the very light end while you're out in the middle of the field. This practical is also a nice opportunity for them to sneak in some more math skills, usually by calculating the overall area of the sample site, or maybe the total area of the quadrats you sampled.